Welcome to the Proteomics course. In previous lectures, we talked about two-dimensional electrophoresis workflow, how to perform two-dimensional electrophoresis step by step. We discussed the protein extraction, we discussed about isoelectric focusing, equilibration, second dimension separation by using SDS page. Finally, we talked about different type of staining methods which are available and how to scan and analyze those images. We then further talked about different advanced methods which are creative ways of overcoming limitations of gel based approaches. Now, in the same theme, today we will talk about another advanced method of gel based proteomics, difference in gel electrophoresis or DIGE, which is used for overcoming various limitations of traditional two-dimensional electrophoresis. So, the previous lectures focused on gel based proteomics, the 2DE workflow and the new methods which are used for proteomics applications. In today's lecture, we will talk about different challenges which are associated with two-dimensional electrophoresis and how to overcome those challenges by using another advanced method, 2D fluorescence difference gel electrophoresis or DIGE. Now, in the previous lectures, when we talked about obtaining a good 2D gel image, then you may realize or feel that at the end of that experiment, all the 2D gel image will look very good. So, I am showing you one 2D gel which is appearing very good, and this is something when everything goes very well. So, if your protein separation and staining everything is fine, then at the end you should be able to see very good protein separation on the gel. This is a representative 2D gel image shown with the human serum sample. Now, I am showing you another good gel obtained from bacterial sample, the B subtilis, but often this is not the case. Many times there are different type of issues which could give rise to very bad images. These issues could be because of the sample preparation method, because of reagents involved, because of isoelectric focusing, different type of parameters involved, and finally, different type of staining methods being used. So, let us talk about some of these issues step by step. So, there are various challenges which are associated with two-dimensional electrophoresis. Mainly, the gel artifacts are major limitations. How to compare two gels, how to minimize the variations. Often during the electrophoretic run, there is lot of run to run variation. Then you have different type of technical and biological variations. The image analysis, that itself is a very challenging process. How to define the spot boundaries, how to extract the intensity information from those spots. Often there is a user bias in doing this type of data analysis. So, there are major sources of variations in two-dimensional electrophoresis. I am showing you few problematic gels, how even the small mistakes or some issues inherent to your biological samples can give rise to very bad 2D gels. For example, I am showing you few sample preparation issues. In this gel, as you can see, the TC acetone precipitation method was followed, but there was not good washing performed. So, TCA still remained in the pellet and one can obtain the pattern as shown in this gel. Now, if you are performing the planned protein extraction, but the solubilization is not sufficient without precipitation, one can see these type of streaking and uneven gel pattern. Now, if you have samples which are having proteins in the abundance, such as crude serum sample. So, these samples will show you different interference in the focusing strip because of large salt and other interfering components present, and then you can see these type of bad gels because of the either salt or different type of abundant proteins present in the mixture. Now, the sample variation is one problem. Often the chemical impurities or the chemical ingredients can also give rise to different type of bad gels, such as the chemical impurities shown in this gel, where the urea is very impure and one can see the carbamylation trail as shown in this gel. If the timid is very old, often the timid is very small amount of timid is used in making the second dimension gel SDS page. So, people often use that bottle for very long time, and if timid is very old, you can see some bad pattern on the gel, something similar to as shown in this image. Now, if your chemical such as tris is not very good quality, it is impure, again that will show some artifacts in the gel. Streaking, that is very oftenly seen because of different type of problems which could be associated, whether it is coming from the sample preparation or because of different type of interfering components. Often you have nucleic acid or carbohydrates is still present in the mixture. The extraction method was not sufficient to eliminate all of those impurities. Many times salts and different other interfering components are also present, which interfere in the IF process, and one can see the streaking pattern after looking at the staining of the gels. So, as shown in this gel, the presence of salt can be interfering and may result into vertical streaking as shown in this gel. If you are using narrow pH gradient strip such as 4 to 7 pH strip, then the vertical streaking can appear, because all the proteins beyond 7 pi will stack together in that region and one can see the vertical streaking. Now, abundant proteins, as we briefly talked last time, sometimes crude samples do contain highly abundant proteins and salts. So, abundant proteins are one of the major interfering components which can be easily seen on the gel. If you have serum sample, there are many interfering components present in the serum, including some abundant proteins such as serum albumin. Now, in the plants, one can expect rubisco as one of the very large protein present in the plant leaves. So, these abundant proteins, they mask many smaller protein and create problem in doing the two dimensional electrophoresis because of IEF process, as well as when you stain on the gel, you can see different type of streaking, as well as different, the whole region is masked because of the abundance of that protein. So, in this gel, it is shown that the human serum protein contains high abundant proteins, which is visible in this area. Now, there are various ways one can overcome these limitations, and as in the previous class of the protein extraction and sample preparation, we have talked about how to overcome these limitations, how to remove the highly abundant proteins. So, please refer to that lecture and one can overcome this limitation. Now, after protein extraction and IEF process is done, one needs to equilibrate this strip. Equilibration process itself can create some problems. For example, if you have performed only first equilibration with DTT and forgot to do the second dimension, the second equilibration prior to doing the second dimension separation, then you can see some pattern similar to as shown here, that only equilibration with the DTT. Many times people think that by increasing the time of doing equilibration, probably the gel quality may appear better, but that is not the case. In this gel, it is shown that by increasing the equilibration time to 20 minutes of DTT and 30 minutes of iodosamide, the gel pattern looks very problematic, and that is because of the equilibration excess. Now, IF process is very crucial. Many times during the IF run itself, one can monitor the problems if you have a software which is showing you how the run is progressing. Often, if your voltage setting is not correct, it is insufficient for the complete focusing, it may result into the under focusing, as shown in this image here for the under focusing. Now, if you have run too much voltage and the overall volt hour is very high, then it may result into over focusing. So, optimizing a focusing protocol for the appropriate duration and overall volt hour is very important. So, there are different type of sorts of variations can be introduced from the biological as well as technical means. There are different biological variations on which there is no control. If you are doing some clinical studies, then even if you have done proper age and gender match, still there will be some variations due to each individual's uniqueness. So, those biological variations are very tough to eliminate, but then there are a lot of technical variations which can be improved during the sample preparation, during the IF settings, during equilibration steps. So, all of those steps may result into large variations. So, in 2-dimensional electrophoresis, when you are separating your control and treatment gels on 2 different gels and you have so many variations coming from both biological and technical variants, then overall the analysis and the confidence in the data becomes very questionable. To overcome those limitations, the new approach, the advanced electrophoresis method, 2-dimensional difference gel electrophoresis or 2D DIGE have emerged. The 2D DIGE method was basically aimed to overcome the gel to gel variability, which was observed in the 2-dimensional electrophoresis, and it allowed the multiplexing samples. This method was first reported by Unlu et al. In 1997, and this method finally became very user friendly, and now thousands of laboratories worldwide are using this technology to overcome the gel to gel variations encountered by traditional or conventional 2-dimensional electrophoresis. So, this method, the DIGE method, involves protein labeling and separation techniques. The optical detection of the proteins is done by using the fluorescent tags, and this method performs the linear detection in the wide range of the protein abundance. So, overall one can expect a very sensitive method for giving rise to very large dynamic range of the protein detection by using fluorescent tags, and this method is giving the multiplexing and separating proteins on one gel. So, it is expected that it will avoid lot of difficulties which are encountered in the traditional 2D gels. So, let us talk briefly about a basic principle involved in DIGE technology. So, we have to discuss how to do the protein labeling, then what is the need for doing the internal standards. So, let us talk step by step. As we discussed, this is the commonly used fluorescent protein labeling reagent for the DIGE. The N-hydroxysuccinamide or NHS ester derivatives of cyanine dyes such as Cy2, Cy3 and Cy5 are used in this method. The Cy3 and Cy5 are used to label your control and your treatments. One should always try to do the dye swapping, so that there is no dye bias. So, if in one sample you are adding the psi 3 in control and psi 5 in treatment, in other sample you should do the vice versa. So, the psi 2 remains consistent for doing the internal standard. Now, for psi 3 and psi 5, these dyes the size and charge is matched, so that the label samples can co-migrate within the gel. Now, I will talk about the labeling methods used in DIGE technology. So, when we are talking about two-dimensional electrophoresis workflow in the last class, we had looked at different type of staining methods currently available, such as Kumasi, silver staining, Cypro ruby, and in that context I briefly described about cyanine dyes. So, now in this technology, the DIGE technology, those cyanine dyes are used. There are 3 different psi dyes, as I mentioned, the psi 3, psi 5 and psi 2. The excitation and emission wavelength of these 3 are defined. The psi 3 excitation wavelength is at 540 nanometers, emission wavelength at 590 nanometers. The psi 5 excitation at 620 nanometers and emission at 680 nanometers. The psi 2 excitation at 480 nanometers and emission at 530 nanometers. So, these dyes are photostable, they are pH insensitive and spectrally distinct. By using these dyes, this DIGE method can be used. Now, let us talk about different type of labeling methods. In DIGE, there are 2 different type of labeling methods which are possible. One is most commonly used, the lysine labeling, or other is cysteine labeling. Lysine labeling method, also known as the minimum labeling method, where the charge and size of dyes are matched, there are no multiple labels, and it is known as minimal labeling because it is only labeling the 3% of all the proteins. I will give you the reason why that is. We are aiming for very small amount of proteome to be labeled, and that will be covered in the next slide. The saturation labeling is done by using cysteine tagging. So, amino acid cysteine is targeted to label. This is charge neutral and size matched. Multiple labels are used. It is very sensitive and used only for the psi 3 and psi 5 labeling. So, saturation labeling is not so widely used, but this is another method one can use for labeling the proteins, and it can be used in DIGE methodology. So, let us talk about the popular method of targeting lysine. So, why to label only lysine amino acid? Lysine is preferred amino acid, as I have shown you in this graph here, the distribution of amino acids across the proteins and their percentage distribution. So, lysine is preferred amino acid to label, its frequency is greater than 5%. So, statistically almost every protein has at least one lysine residue. So, there is a good probability that your protein will have lysine residue and it will get labeled if you are targeting the lysine. But if that is not the case, one can always try the saturation labeling of cysteines. Now, let us talk in more detail about the minimal labeling or lysine labeling. So, psi dyes, they react with the primary amino group of target proteins, and the N terminal alpha amino group and the lysine epsilon amino groups, they are labeled by the process of nucleophilic substitution. So, all the available lysine labeling would create a very hydrophobic environment of the proteins. That was the case observed by the original paper derived by Unlu et al. So, to avoid that hydrophobic protein atmosphere, it is recommended to do the minimal labeling of only 3% of the whole proteome. So that overall it is not very hydrophobic. Now, in this slide I will describe you the labeling chemistry. So, as you can see in the structure here, the structure of Si-difluor comprised of a positive charged Si-difluorophore, a linker and NHS ester reactive group. So, at pH 8.5, the NHS ester couples with amino group of lysine in the proteins and it forms a covalent bond. The amino group of lysine residues, they are positively charged at neutral or acidic isoelectric points. Di is also positively charged. So, the net pi will be unchanged. Therefore, the psi 3 and psi 5, these are not going to alter any mass as well as charge overall, and they are very well spectrally resolved. So, people prefer doing lysine labeling. It is not going to alter overall pi values. It is not going to alter overall mass, because both psi 3 and psi 5 will carry the same mass of these labels, and there is a good probability that your protein will have lysine residues and it will get labeled because of the cyanine dyes. So, how to prepare the sample to perform the DIGE experiment? First of all, you need to reconstitute your dye in dimethyl formamide or DMF. The lysis buffer, that is in which your protein sample is going to be dissolved, consists of 30 millimolar tris, 7 molar urea, 2 molar of thiourea, 4% of chaps, and you have to adjust the pH at 8.5. Please make sure there is no IPG buffer or carrier ampholyte, and there is no reductant added in this lysis buffer. Since you want to perform the minimal labeling, you need to add only 400 picomole of Si dyes in a small amount of protein, 50 microgram of proteins, and during that process the reaction should be performed on ice water for half an hour. If you go back and remember about our previous discussion, to visualize a gel with the Kumasi stain, you need more protein. Even from silver and other stains, you need good amount of protein, but these fluorescence dyes are very sensitive. So, all you are looking for a very small amount of protein, 50 microgram protein quantity is sufficient for performing this experiment. Now, if this reaction of half an hour is done, after that reaction should be quenched with excess of primary amine, the free lysine, by adding 1 microliter of 10 millimolar of lysine. So, then reaction will be stopped and you can proceed with the next step. So, while you are doing this reconstitution of dyes and labeling your protein samples with the Psi 3 and Psi 5 dyes, now let us think about our Psi 2 dye and the internal standard. So, I am taking a very simple case here, where you have 4 healthy controls and 4 patients suffering from a disease. So, to represent all the proteins which are present in each of these samples in healthy individuals and diseased patients, first you need to make an internal pool which is representative of all of these 8 samples. So, you can take 25 microliter from each of these 8 samples and make it to 200 microliter of one pooled sample, which is your internal pooled standard. So, this pooled standard preparation can be performed by combining the aliquots of equal amount of protein from each samples in the given experiment. Now, the Psi 2 dye you are going to label with this pooled sample, so that can be used as a internal standard. This internal standard has many advantages, and this is actually one of the major success that how 2D limitations have been overcome. Since every protein is present in the internal pool population, so that is going to be appearing on your gel. So, there is a reference point for each gel already present on your gel, whether it is from any patient or any controls. So, the gel to gel variation will be reduced. Now, gel to gel matching and comparison will be much efficient and easier. Now, this internal standard is creating a reference point for every protein on each gel in the experiment. Later on, when software performs some data analysis, this results into accurate spot statistics and more meaningful biological data can be interpreted from this analysis. So, now we have talked about the DIGE principle, how to label the samples with different psi dyes, why there is a need for making the internal standard, and how to label the internal standard with the psi 2 dyes. So, let us go to the step by step procedure of DIGE method. So, very first thing is you have extracted your protein samples and you need to make the internal standards. Now, your control sample, treatment sample and internal standards should be labeled with different cyanine dyes. Internal standard is always fixed with psi 2 dyes, but your control and treatment should be dye swapped. So, sometime it should be psi 3 and then few samples should be psi 5 and the vice versa. Once labeling is done, then sample should be mixed. So, all the 3 samples psi 3, psi 5 and psi 2, all 3 labeled sample should be mixed in one combined tube. After that process is done, you can take that sample and do the rehydration. We have talked about rehydration step and the need for it in the previous lecture in the 2D workflow. Now, this rehydrated strip can be focused in an isoelectric focusing instrument. It is good idea to avoid any light exposure during the rehydration, dye reconstitution process as well as during the isoelectric focusing run is in progress. Once IF is done, again you have to do the equilibration, first equilibration and second equilibration followed by SDS page. So, overall step as you can see, it is exactly same what one is doing in the 2-dimensional electrophoresis. Only variation here is that all your control and treatment samples have been mixed together and now they are separated on one IPG strip. Now, this strip is placed on one SDS page gel and all the proteins will be separated in one gel itself, whether it is coming from the control or treatments. Now, this one gel can be scanned by using fluorescence scanners such as the one shown here in the DIGE imager, and psi 3, psi 5 and psi 2 patterns can be obtained. So, this slide gives you an overview of the whole DIGE process, where one needs to mix psi 3, psi 5 and psi 2 dyes in one tube, resolve those on one gel on SDS page and then scan that one gel with different excitation and emission wavelengths to obtain 3 gel images derived from the same gel. Here few representative gels are shown obtained from the DIGE experiments for psi 3, psi 5 and psi 2. Software also generates the combined image obtained from mixing all the 3 dyes and the overlapped image, which shows you the abundance of certain proteins. The red and the green are indicative here. In overlapped image, the red and green are indicative of over and under expression or upregulation and downregulation, and the white or yellowish color is representing that there is no change. So, DIGE image analysis, although it is very software driven process, it is much more automated as compared to 2D analysis, but it requires lot of tools which can be used for good interpretation. So, from your one gel you have obtained 3 images now, and those can be further analyzed by using DIA in the co-detection of 3 images. So, I am talking about a software decider which takes these 3 images in account and makes one image pool from it by the process of DIA. Now, these images, the multiple DIAs can be matched in the BVA set. So, these BVAs can then further be used for obtaining statistical information about these protein changes, whether those are statistically significant or not. So, I am showing a representative gel here, where one spot is highlighted, in fact 2 spots are highlighted, shown in disease and healthy controls. So, one can look for the abundance of those proteins in the 3-dimensional views, and as shown here, if these proteins are showing higher abundance, those are visible in different treatments, whereas there is no protein change happening in the healthy control. Now, one need to look at the same trend of these 3-dimensional views across all the patients. So, for example, I have selected here the group of 6 patients and 6 healthy control. Now, this particular protein is showing upregulation in all the 6 patient, as you can see here, and there was no change in this protein level in all the healthy individuals. So, by looking at the data carefully patient wise and the reproducibility pattern, one can be convinced that this protein is having some biological relevance and then need to be further identified and characterized. So, after doing the BV analysis, further statistical analysis of different classes can be performed by using EDA or extended data analysis. Now, EDA has different type of tools in this software. One of the most commonly used is principal component analysis or PCA. The principal component is a linear combination of the optimally weighted observed variables. Now, PCA analysis is used to test whether in your protein samples in different patients and controls, the protein expression is uniform across all the multiple samples derived from the same experimental group. So, if your results are consistent across the whole population, the same protein pattern is consistent in all the controls and in all the treatments, then the spot map from the same experimental group will be located in the same region. To clarify this point, I will show you this image here, the next slide. If you have one control and 2 treatments, and by looking at the significant proteins, the software can use this tool, principal component analysis tool, and give rise to the pattern of these healthy and different type of treatment patients, and if the grouping is together, it means it is highly significant. As you can see here, the red dots are showing the controls and blue and yellowish dots are showing 2 different type of treatment groups. So, now these 3 groups are clustered quite closely. There will be some heterogeneity because of the different clinical samples and patients biological variations involved, but overall it shows that data is quite statistically significant in the experimental groups. Now, by using this software, one can obtain further information about different type of discriminant and classes. So, the discriminant analysis is used to classify the uniform data for the known classes. One can initially take all the proteins which are significant and then try to group those based on the known classes. The hierarchical clustering is another method which is giving very useful information. For example, in this data we try to look at one control and two different type of treatment groups together. Now, on the extreme right side, all the green shades are from the healthy controls, and then there are two more population from the patient's group, which are showing many proteins in the red region. It means it has higher protein abundance. So, this clustering is showing that the control is belonging to one population, and these two treatment groups are derived from same population, but even within that population they have certain variation. So, these items, whether proteins, spot maps or experimental groups, can be grouped together in the hierarchical way by using this clustering. So, now I will describe you the animation process, how to perform DIGE experiment, and you will be able to recall different concepts which we have discussed so far. Difference gel electrophoresis, DIGE. The pooled internal standard for DIGE is prepared by mixing the equal amount of all the samples that are being run in a given experiment. The internal standard prevents problem of gel to gel variations. Now, let us talk about dye labeling. Each protein sample as well as internal standard is labeled with a differently fluorescing cyanine dye, which allows all protein samples to be simultaneously run on a single gel. This dye binds covalently to the epsilon amino group of lysine residues in protein. These samples should be mixed together, and labeled proteins can then be run on a single 2D gel. The separation takes place on the basis of isoelectric focusing in first dimension, followed by the molecular weight separation in the second dimension. The gels containing all the protein samples can be viewed by illuminating it alternately with excitation wavelengths corresponding to psi 2, psi 3 and psi 5 dyes. Now, the superimposed 2D DIGE image can be viewed. Information for the molecular weight and isoelectric point can be obtained from the gel. But most importantly, by looking at the gel pattern, if you are doing the abundance proteomics, comparing the expression of two samples, different protein expression, then that pattern can be immediately observed on the gel with the red and green patterns. If there is no change, it will appear as white. Okay, so after knowing the concepts and after looking at the animation, now you should be very confident in overall concepts related to the DIGE experiments. So, let me give you a very simple problem. If you want to perform some gel based proteomics approach and you have been given 250 patients from a clinical trial. So, which of the two methods will be useful, whether you would like to use 2D electrophoresis or difference electrophoresis DIGE. So, which of these two methods would be better technique to separate serum protein samples obtained from 200 patients in a given clinical trial. So, let me give you this interactivity and ask your answer. So, if your answer is 2D gel electrophoresis, that is not correct. You need some method which can minimize the variations across large population. So, you need difference gel electrophoresis. So, let me describe you this concept in the animation in the following slide. So, two-dimensional electrophoresis, although it is a very useful technique, but may not be best option in this case for analysis of serum proteins obtained from large number of patients, as it would involve running several individual gels, which would be a very time consuming process. Also, there will be variations across the gels, would make comparison very tedious. Whereas, the DIGE can be extremely valuable tool for the analysis of large number of samples simultaneously without having to overcome the problem of gel to gel variations. The control and test samples can be differentially labeled by using side dyes and run on a single gel. Finally, you can scan the gels by using a DIGE imager. Okay. So, now you have a good clarity that what are the disadvantages of two-dimensional electrophoresis and advantages of DIGE. So, let us summarize our concepts of 2DE and DIGE. So, in 2DE there is gel to gel variations which can be overcome in DIGE method by separating the proteins on one gel. In two-dimensional electrophoresis there is large sample variations, whereas in DIGE this variations can be reduced by making an internal pool or internal standard, which is going to give the reference point for all the proteins. Now, in two-dimensional electrophoresis, often in the data analysis, there is user bias how to define the spot boundaries and obtain the statistical information, whereas in DIGE there is a code detection method involved by the software analysis, which eliminates lot of user bias of performing this analysis. So, overall if you compare 2DE and DIGE, there is definitely more merit to perform DIGE experiments, but not to say that before you want to move towards 2D DIGE approach, first you need to ensure that your sample is good, your technique is working fine, and overall you can obtain a very good gel. So, first you need to perform traditional or conventional 2D gel stain with chroma C or silver stain to see whether everything is looking good, you are able to obtain a very good image, and then you can apply the same protein extract to perform the DIGE experiment. That will be very cost effective. So, in summary, in today's lecture we talked about different challenges which are associated with two-dimensional electrophoresis. Then we looked at DIGE workflow, different concepts involved, as well as how to do the labeling of samples, how to make internal standards, and then we have seen the steps in animation. We finally compared 2D method and DIGE method, and I gave you an interactivity option to perform experiment by using 2DE or DIGE, and you gave the right answer of doing the experiment with DIGE method. So, overall from this whole experiment you should be able to conclude that DIGE is better way of separating proteins, and it can still provide you very useful quantitative information in the gel based proteomics. I would like to acknowledge Dr. Srinivas and Lalit Kishore for providing some useful information related to the 2D DIGE, and we will continue our discussion on 2D DIGE and data analysis in the next class. Thank you.